Hello everyone and welcome to my video lecture series. So today we are going to learn about the monohybrid cross, and along with that we are going to learn the first and second laws of Mendel. So let us start with the topic. First I'm going to show you how the monohybrid cross is done, and then when I have completed the cross I will show you where each law is applicable. So in the monohybrid cross, we are basically using any one character from the 7 characters that Mendel studied, right? So let us take the character of height, and as you all know, each character has a dominant and a recessive trait, right? So height has a dominant trait which is tall, which is represented by the allele capital T, and the recessive trait is dwarf, which is represented by the allele small t, right? Now in the monohybrid cross, both the parents are pure or homozygous. So one of the parents is going to be capital T, the other parent is going to be small t. So let us start with the cross over here. So capital T crossed with small t. Over here I will write down tall, and over here I will write down dwarf. Okay, both these are called as pure parents or homozygous parents because each of the parents has the same allele. Okay, now please remember to denote that these are the parents. Okay, you can also write a capital P over here. Now the next step is to write the gametes. So here I will be writing capital T as the gamete, here small t. So if you want, you can write down capital T over here and small t over here, but since both the gametes form will be same, I'm just writing them once. So also over here please write down that these are gametes. Okay, now the next step is to unite the gametes so that they form the F1 generation, right? And that is capital T small t, and this individual is basically a tall individual, so the phenotype is basically tall. Okay, but the genotype is capital T small t, so it is also called as a heterozygous or a hybrid individual. So I will write down in the bracket hybrid tall. Okay, and over here I am going to write down F1 generation. So F1 generation is the first filial generation which is obtained when both the parents are crossed, or both the pure parents are crossed in the case of a Mendel's cross. So here half of the monohybrid cross is done. Okay, if you want, you can use any other character. For example, you can use the color of the pod, the seed shape, any other character you can use. I have used the height character, and I have used both the dominant and recessive traits, that is tall height and dwarf height. Okay, now the next thing to do is to self the F1 hybrids, that is called as selfing, and selfing can be represented with the help of a Punnett square board or a checkerboard method. So the way to do that is you basically write down the gametes of the hybrid. So capital T small t will have gametes, capital T small t will have gametes. One gamete is capital T, the other is small t. Similarly, selfing is done, so it is self-fertilization. So again the gametes will be capital T small t, and I will draw the checkerboard over here. If you are comfortable doing the cross directly, no need to draw a checkerboard. Now writing the individuals over here, so this individual is going to be capital T, then this will be capital T small t, and small t. So you are getting basically 3 types of individuals based on their genotype, right? I'm going to write them down. The first one is going to be capital T, which is a pure tall plant. The second type of genotype is capital T small t, which is a hybrid or a heterozygous tall plant, and the third one is small t, which is a dwarf plant. You don't need to call a dwarf plant as a pure dwarf because dwarf plant will always have both the recessive alleles, so it is a dwarf plant. So now if we write down the proportion of all these 3 offsprings, the ratio that you get is 1:2:1. Now this ratio I have derived on the basis of the genotype, please remember. Okay, so this ratio is also called as the genotypic ratio. Please remember this. So I will write down over here, this is the genotypic ratio of a monohybrid cross, that is 1:2:1. Is that clear? if somebody asks you what is the phenotypic ratio, then the phenotypic ratio is dependent on the phenotype. So over here in the selfing process, we see only two types of phenotypes, right? So capital T is a tall plant phenotypically, capital T small t is also a tall plant phenotypically. So all those three individuals will be considered as tall plants. So the phenotypic ratio is going to be 3 tall plants, okay, versus 1 dwarf plant. So that is 3 tall versus 1 dwarf, and this ratio is called as the phenotypic ratio. Is this clear? Okay, so that ends our monohybrid clause. Please remember, monohybrid means you are only taking into consideration one character out of the 7 characters studied by Mendel. If I am talking about a dihybrid cross, that means I am going to take into account 2 characters. I will make a video shortly on the dihybrid cross as well, but if you can understand the monohybrid cross, dihybrid cross is very similar. Is that clear? So with that, we have completed the monohybrid cross. Okay, it is very important for you to show the ratios. You have to show the genotypic and the phenotypic ratios. Okay, now coming to the part about the Mendel's laws. Okay, now there are 2 laws of Mendel's which are dependent on the monohybrid cross. Okay, the first law is called as the law of dominance. Now the law of dominance is basically only dependent on this part which I am going to show you over here. So this part which I am marking over here, right up till the point where you get the F1 hybrid, this entire part is related with the first law of Mendel, and that law is called as law of dominance. Now what is the statement of the law of dominance? So basically the law of dominance states that when 2 homozygous individuals, or when 2 homozygous parents, or when 2 pure parents are crossed, the allele which expresses itself in the F1 generation is called as the dominant allele, and the allele which does not express itself in the F1 generation, it is called as the recessive allele. Now first of all, I hope everybody knows what are alleles. 2 or more forms of a single gene are called as allele. For example, if I say that the gene is T, then capital T and small t are 2 forms of the same gene, so they are called as alleles. If I say that the gene is Y, then capital Y and small y are the 2 forms of the gene, so they are called as alleles. Okay, so 2 or more forms of a single gene, they are called as alleles. Now what is the statement of law of dominance? It says that when 2 pure parents are crossed, so we have done that over here. Okay, then what happens is the allele which expresses in F1 generation. Now in the F1 generation, all the plants that you are getting, they are tall, they are hybrid tall, but which allele is expressing itself? Capital T allele is expressing itself in the F1 generation. The small t allele is not expressing, because small t is responsible for dwarfness. But are the F1 plants dwarf? No, they are all tall. So small t is not expressing, so small t becomes recessive. Capital T is expressing itself, so capital T becomes dominant. I hope you are getting this, so I will label it over here. This becomes the recessive allele, and this becomes the dominant allele, and that is what the first law of Mendel says. Okay, so we have completed the first law of Mendel, which is the law of dominance. Okay, and that is the first law. Now coming to the second law. The second law of Mendel also uses the monohybrid cross, but here we have to show the entire monohybrid cross right up till the F2 generation. Please remember, if law of dominance or first law is asked, just show the cross up till the hybrids, no need to self the hybrids. Okay, so coming to the second law now, continuing further, the second law of Mendel basically is also called as law of segregation of alleles. So what I'm going to do is I'm going to write down over here on the left side, since we are having space. Okay, second law of Mendel is called as the law of segregation, or it is also called as law of purity of gametes. I will write down over here, law of purity of gametes. And for this law you have to show the entire monohybrid cross. Please remember, you have to show the F1 generation, you have to self the F1 generation, and you have to show the F2 generation as well. Okay, now what is law of segregation? I will tell you the statement first. So the statement of segregation goes like this, that in the F1 hybrid, when the gametes are formed, the two alleles of the F1, they separate from each other, and each gamete has an individual allele. Okay, so now over here, if you see the F1 hybrid, it is capital T small t, right? Now during gamete formation of capital T small t, here you can see in the checkerboard method, I have shown both the alleles which have separated. Capital T has separated from small t, right? And since it is selfing, again capital T and small t. So what is happening during gamete formation of F1 hybrid? Both capital T and small t, which are alleles, they are separating. And when they are separating, each allele which is separated, that is entering a different gamete, and that is called as law of segregation or law of separation of gametes. It is also called as law of purity of gametes, because the gametes are formed because of separation of alleles. The alleles are not remaining stuck together. See, if you see the hybrid, you will see that capital T small t both are there together, and the dominant allele is showing its effect, but that doesn't mean that they have got stuck to each other. During gamete formation, because there is segregation of alleles, you are getting a individual which is dwarf in the F2 generation. This is only possible if the alleles can separate and not get mixed. That is why it is always said that the gametes are always pure. That means the alleles will always separate from each other. The alleles will remain together in the F1 hybrid, but during gamete formation of the F1 hybrid, they will separate. That is why it is called as law of purity of gametes or law of segregation or separation of alleles. Is that clear? So then when you want to show the second law of Mendel, you have to show the F2 generation, show the checkerboard or the Punnett square method, and please don't forget to write down the genotypic and the phenotypic ratio. Okay, genotypic is 1:2:1. Please write down pure tall, hybrid tall, dwarf, and phenotypic ratio is 3:1 tallest to dwarf. Okay, so that ends our today's part of this mini lecture. If you like the lecture, please put a thumbs up, leave a comment, subscribe to my channel, press the bell icon, and you can share this video with your friends. Okay, in the next lecture, I am going to talk about the dihybrid cross and the third law of Mendel. Thanks a lot.